So this is called the pseudoheart. That's basically pumping the blood around. And it's red. It has hemoglobin. It's not within red blood cells. It's freely in the lipid portion of the blood. Okay. One semester with a student, it was actually the UGSI. They saw the blood in the earthworm and they hit the ground. So just realize if you feel faint, put your head down between your knees, go out and get some fresh air. Okay. Because we really don't want to have major injuries in lab. Here's the GI tract. Now we have a true tract. So we have a true mouth and a true anus. And as we go through here, we have specialization. So one specialization is the pharynx. It's much more obvious in the earthworm. The diagram here is really not very impressive. So we have a pharynx. We have esophagus that leads to a crop for storage. We then have a gizzard for fragmentation. Again, this diagram makes it look very similar. I'll show you some images shortly. It's really easy to tell these various parts apart because the pharynx is highly muscularized, the crop is large, and the gizzard you're going to see bands of muscle. And then the intestine out and then the rest of the anus. Okay? This structure here is meninferidium. That's for nitrogenous waste and osmotic balance. So here they're called auxiliary hearts. You don't have that image, but it just illustrates again the pseudohearts. Here shows you one of these segments. Some terms that confuse students sometimes, which is completely understandable. A somite is a segment. So a segment is a somite, and a somite is defined by two septa. In your nose, you have a septum that divides your two nostrils. So we have a septum here, and another septum there. That's one segment or somite. Okay. So this septum is the membrane that partitions these segments. And the beauty of this is it allows for this earthworm to create a hydrostatic skeleton because you have two segments like this, right? The two septum. So if you contract the muscles, they have two types of muscles. They have longitudinal muscles that go along the length of the earthworm. So if you contract the longitudinal muscles, and I didn't bring a balloon, I should have. If I were to contract longitudinal muscles and it was a balloon, I'd be doing this, right? So the balloon would get shorter but fatter, right? And if I took and contracted circular muscles that were on the circumference of the balloon, I would squeeze it and the balloon would elongate, right? So you can see that the shape of one of these segments can vary. Well, that's not very helpful except that they have these structures out here called setae that they anchor into the soil. So if they elongate, so from here to here, and they anchor the setae, when they constrict like this, they contract the circular muscles, the longitudinal, they'll get shorter here and they will move. So basically they can move forward by alternating contraction of those muscles along with the anchoring of those setae, okay? So this just shows you one of those meninferidia. Notice there's an opening here in the coelom. This is a true coelom, a cavity. So we have the outside body wall, we have the GI tract, we have peritoneum here, peritoneum not shown here, but there's peritoneum on here as well, and we have this cavity, the coelom. So coelomic fluid gets pulled into this tube. Once it's inside that tube, it's outside the body effectively, it passes through and eventually is released. Now you see these capillary beds surrounding this tube. Why would you have those capillary beds? What would be the purpose of those capillary beds surrounding those meninferidia? What's happening in those tubes? Diffusion. We're actively taking things up into the body, and so they get taken up into the body by these capillary beds. The exact same thing's going to be true for the kidney. Okay, they're called vasa recta, but anyway, it gets taken up into the capillary beds. We also have to have secretion, right? So we have exchange between the capillary beds along these tubes to actively secrete into the tube. Okay, and then just here's a cross section here. Now this is not accurate because you'd never find the opening here called the nephrostome in the same segment as the nephridiopore. This would be in a different segment, so that part's inaccurate. It's just trying to illustrate some things. Here you can see this thin layer of peritoneum here, mesodermally derived, and notice it comes down and covers the GI tract. Okay, the GI tract, the intestine here has an infolding called the testisol. What might that infolding do? Increase surface area for exchange of nutrients, uptake. Okay, so and then this is the dorsal blood vessel here. Here's a ventral nerve cord here. It's going to appear white, and there's a ventral blood vessel. Okay, so in a slide, cross-section, an image, you can tell me what's dorsal, what's ventral. Okay, you can't tell me what's right and what's left because you don't know if I have that oriented like this or this, but you know what's dorsal and what's ventral. And here's to show you these setae sticking out. They help to anchor. And we should have movies of this. You actually see the movie how these things go in and out. And we have live earthworms in the lab. So if you want to take that earthworm and put it in your hand and then pull the earthworm's trying to overcome your pulling, and they're trying to stick their setae out, and they're not going to anchor their skin. You don't have to worry about that. But you'll feel a little grip. You can feel them that you can't pull it too well. Okay, if that's not sensitive, try the back of your hand because that's more sensitive than your cheek. And what's even more sensitive? The tongue. But don't do that one. I'm going to tell you again, don't do the tongue. Not in Bio 1A. You do it elsewhere if you want, but not in Bio 1A, okay? I got in trouble last semester. All right? Don't use your tongue. It is more sensitive, but just use the back of your hand, okay? Or your cheek's fine. Here shows you the section. Here's the pharynx, highly muscularized. Here's the esophagus. There's those pseudohearts. There's that dorsal blood vessel. There. Here's the crop for storage. The gizzard, highly muscularized. Esophagus here. This white structure there is the ventral nerve cord. And these structures here along the side, those are the septums. The septa have been cut. When you pull that open and pin it to the side, you're basically ripping those septa, so you can pin it out. Here's a close-up. The crop is here, it's been cut. See how highly muscularized this gizzard is? That's because when the earthworm moves through the soil, it basically moves with its mouth open, and it's just basically taking in soil. And hopefully there's leaf debris in there and other organisms, nematodes, viruses, etc., whatever it could potentially feed on, and then it takes it in. But it has no teeth, so it can't fragment the food, but as it crawls through the soil, there's bits of sand. And what happens is, since the gizzard basically is very muscularized, it goes back and forth squeezing, like when you knead bread, it squeezes, and those pieces of sand get basically pushed against one another, and if there's any leaf debris, etc., it fragments that. It's like stone ground wheat is wheat berries between two large stone wheels, and the weightless two wheels, they spin it, and that basically fragments the wheat berries, so you get the flour. In this case, they don't have such an idealized structure. They have those sand randomly, okay? So that's the purpose of the gizzard, highly muscularized. Fragments the food so that as it passes through the GI tract, digestion can occur, and then absorption. And they have this specialized yellow tissue here on the GI, on the intestine, called chlorogoginous tissue. Chlorogoginous, it's liver-like in function. So on the exam, if we ask you what's an analogous structure to liver in an earthworm, you say chlorogoginous tissues. It stores glycogen, like our liver. It does formation of urea, like our liver, and it does some detoxification. Okay. Here it shows you the anterior end of the earthworm. Here's that pharynx, highly muscularized. There's all these connections here. Notice they connect to the internal body wall and to the septa because it has to anchor. The pharynx has to develop a force to pull food in. So that's why you have all these anchors. We anchor muscles to bones, etc., to create leverage. In this case, they don't have that bone, so they attach it here. Here's what's called the supra above, pharyngeal, because it's above the pharynx, ganglia, clumping of neurons. Okay? Again, at the anterior end. Here's those pseudohearts. Here's some seminal vesicles here. These are those structures called spermatheca that store sperm that were donated by the other worm. Okay? So this is where they store the sperm. The testes are within these seminal vesicles, so we really don't see them. There's an anterior, middle, and posterior. Don't see the posterior there. Okay? But you'll do the race sections here. We can see this. Here's one of those walls, You can barely tell this, but there's a structure here that floats. And if you look carefully, there's red right there, a capillary bed. This is example of one of those meninferidia. Here's the septum there, that body wall, but here's a meninferidium there. And those meninferidia and clearly the septa are repeated throughout the body. Okay? So when you do the worm, you'll see that in this section. Here it just shows you this infolding that's been cut open. And there we can see food debris basically in there. Okay, and then this is a polychaete. It doesn't have setae that stick out, but it has these structures called parapodia, and they help with gas exchange. And this is a feather duster, pretty bizarre looking, but indeed it's an annelid, and it's a polychaete, and these are modified parapodia for basically filtering food and some gas exchange. And this is an article I got a few years ago from the paper. Basically, in northern Minnesota, various parts of northern U.S. and Canada, there's different earthworms that have been brought in. And because these earthworms have a different impact on the soil topology is changing in terms of the tightness of the soil net, and the trees are actually dying off. So you have huge forests dying off because the earthworms that are there now are outcompeting the native earthworms and changing the porosity of the soil, which affects the ability of those trees to grow. So there's lots of interest about this. This is one reason why when you come into California, they have that agricultural check station. They're supposed to look for plants and things like that, soil. You're not supposed to bring in potted plants because potentially earthworms, new earthworms, or nematodes, or various diseases like that. That's why they have those egg check stations, especially in California because we make so much money off the agricultural sector. In terms of the mollusks, 4 classes we're going to look at, the Polyplacophora or chitons. They have 8 plates. They're pretty easy to tell. The gastropods are snails. One group of these are the nudibranchs or naked snails. They're usually brightly colored because they have toxins within them. And the reason why they have toxins is not because they make them, but they feed on these cnidarians. So those nudibranchs can feed on cnidarians. Amazingly enough, they feed on cnidarians, and their GI tract has evolved so that they don't digest those nematocysts and that. They actually take them up, and then they store them in their own body wall, so that when a predator tries to feed on them, they get the toxins from their stored nematocysts. So they don't make them, they get them from their food source, and because of that, they're highly colored to help warn predators. Bivalves, another class, that's the demo we'll be doing, the clam, and cephalopods, squid, and octopi. And then this illustrates 5 features of mollusks. They have a mantle that secretes the shell. So when you look at the clam demo, you'll see the mantle and the shell around it. A pearl is typically usually an irritation of the mantle, sand grain, whatever, and they wall it off with the shell, and that forms the pearl. Okay? Some species are better than others. Cultured pearls, in case you're wondering, they put pieces of glass beads that define size in there, so those pearls are very uniform and round. And, of course, glass in the middle. Shells, that outside shell. A foot for movement. Cnidia, which are these gill-like structures for gas exchange and filtering food. That's why they're not called gills. And a radula for scraping food. Now, the bivalves, because they're filter feeders, they've lost the radula during evolution. So when you look at these groups and the mollusks, we have the polyplacophorans, or the chitons. We then have bivalve. And then, I don't have enough space to write it, so I'm just going to draw it here. We have the cephalopoda and the gastropoda. So these two groups are more closely related than these two groups. Okay? So I'm just going to write C and G, so you know that's the cephalopoda there and gastropoda. And remember, you can flip this either way. It still maintains the same relationship, okay? And I have been known to just put cladograms, 5 cladograms on the lab exam and say, which one is different than the other? I don't put any groups on it. I just draw 5 different cladograms, and you should be able to tell, well, if I flip it this way, it looks just like this, okay? This is from the lab manual. Occasionally asked, what do you mean by derived versus ancestral feature? Ancestral features are features that groups lower on the cladogram have as well. So 3 germ layers would not be a unique feature or derived feature for the mollusk. But things like a mantle and foot and radulence and the shell are. But things like the coelom, well, we saw that in other organisms. Segmentation, we certainly saw that in the segmented worm. Cephalization, we saw that. So those are ancestral features. Okay? And that's from the lab manual. It's just a table, table 1 in this IVERT 1 lab. In terms of mollusks, again, this chart you can go through and fill out. This is just some illustrations. Here's those polyplacophorans, those plate-like structures. Here's an example of a snail and a snail. This is the nudibranch. Land snail. We'll have snails in lab for you to look at feeding. Basically, put some cornstarch on a petri plate, and we have a mirror there so you can actually watch them. And they literally take their radula and scrape the cornstarch. And it's kind of a black structure. We have the radula set up in a microscope. So you can look at the microscope, see it looks just like a file, but then watch the live snails feeding, and you'll see this radula go back and forth. If you ever have the opportunity and you're hiking in a forest like down Santa Cruz and that, and you come across snails feeding on a mushroom, it's great to watch them because the mushroom is three-dimensional. You can literally watch them shave off layer by layer of that mushroom as they feed. Cephalopods here, squid and octopi, the nautilus here. This is quite interesting. They're fairly intelligent. There are documented example of where there's these 2 marine tanks set up, and they have fish in this tank, and the fish kept disappearing. I couldn't figure out what was going on. So they have documentation of a octopus crawling out of one tank, going over to the other tank to feed. But because there's no overhang or shelter here, it, of course, felt more prone to predation. So it actually literally crawled back to the other tank and went under ledge for protection. And they actually have observed this with the camera. So pretty complicated behavior. It's used for various studies like that. They have giant neurons. Anyway, here's a chitin here. 8 plates, 2, 4, 6, 8. Here's a nudibranch here. Here's just another gastropod here with a shell, of course. Here shows you a squid here. They basically constrict their mantle to shoot off water. A lot of work being done on squid and behavior. This is maternal behavior. This is an egg mass here, filled with oxygen so it can get gas exchange. When it's threatened, it basically compresses this and then jets off for protection of the egg mass. Things like, there's a beak here that they use to rip apart the food when they feed. And they're looking at the attachment, and they're trying to figure out how it's attached and the tissue there, and it can potentially help with new prosthetic devices about how you attach those to minimize damage to the attachment point. This is from a professor here, talking about how they basically mate. Here's a scallop here. Here just shows you the shells here. There's a right and left side, so there's bilateral symmetry. So you should be able to tell what's the right shell or the left shell. So I want to go over that. This here is the right shell. So the way you're going to know this is, if you look at this, there's a foot. So this organ is going to move, in this case, to the left as you view it. And then here's the shell or bivalve here. There's the dorsal part here, and there's the elastic hinge. And if you look carefully, the distance here, anterior, is shorter than the distance posterior. So when you look at the shell without the organism, we see here the dorsal surface, ventral. This is the umbo, the part where it starts growing from initially. See how short this distance is? This is anterior. In this case, we move to the right. This is posterior. So this is the right shell, makes this the left shell. In fact, on the shell, you'd see a scar where the mantle used to be attached. Plus, they have muscles called adductor muscles that close the shell. So adductor muscles add to midline. So when you go to the gym and you're in the exercise machine, and with your legs you're trying to push them together, you're exercising your adductor muscles. If you do the machine where it's this way, you're exercising your abductor. So adductors add to midline. That closes the shell. So if you had a starfish trying to feed on this clam, it would be going there trying to open up the clam, and the clam would use its adductor muscles to close up. We're going to talk about filter feeding here. Always confusing. These have an open circulatory system, but let's go through feeding. They have a in-current siphon where fluid gets pulled in, and it gets pulled into a cavity called the mantle cavity. So this is going to be best illustrated actually looking at the clam. So here's the mantle cavity. It's not a coelom. The coulomb would be inside the body mass here. So we have an in-current siphon here. It sticks out. There are two. It's two tubes basically stuck together. We're looking from the ventral surface. Here's the structure. It's called the tenidia, the gill-like structures that do gas exchange and filter feeding. There are cilia, the small structures on the tenidia that beat. They create a current. Just like if you're in a bathtub, with your right hand, you do this, the current moves around, right? You can put a filter there. So when the cilia beat, they create a specific current. They close off this cavity when they're filter feeding. So the cavity's closed off, so the only way fluid to enter is through this in-current siphon, which is ventral, goes past the tenidia, and then goes out the excurrent siphon, which is dorsal. We can't see the excurrent siphon. So fluid comes in, passes, comes into the in-current siphon, enters the mantle cavity, goes through the tenidia, out the excurrent siphon. Fluid gets captured, and the fluid gets transferred on the tenidia up to the mouth, and there are structures called labial pelts that help transfer that. Enters the mouth, goes to the GI tract, and then the waste would be released to the anus. Okay? So what we can see here, this is the foot, so does it move to the right or to the left? Foot here, moves to the right, it moves in this direction. That made this anterior, that makes this posterior, ventral, dorsal. Here, if you've ever eaten scallops or seen scallops, basically scallops they have a huge adductor muscle, and you see the scar, that they use to close the shells. So this here is the anterior adductor muscle, and the posterior is dorsal, we can't see it. So anterior adductor, here's the mantle edge here, here's the shell or bivalve there. Here's from the dorsal aspect. We've cut this excurrent siphon. Normally this would all be closed, you could not see in there. So it's once that mantle cavity is closed, it's like a closed system with an opening incurrent and opening excurrent. So we've cut this, and what we see now is the back of the tenidia from the dorsal aspect. So we know fluid has to go through there in this direction. I'm not going to use the pointer, I don't want to blind anyone. Goes through this direction, up towards you, and then out the excurrent siphon. And as it does that, it gets captured by the tenidia, and fluid gets transferred to the mouth. Waste just get released, and then any waste from the anus, the anus here gets dumped into the excurrent siphon. And again, this is closed up. So what you're doing then is you're dumping your waste out the excurrent siphon. Likewise, gametes, upstream or downstream of the filter. Downstream, if they're upstream, you'd be filtering out your own gametes, would have been a waste to make them, right? So you dump your gametes downstream of the tenidia into the excurrent siphon, okay? And you can't see this clearly here, but there's a very small heart here that's brightly orange colored, and clams don't have a really high metabolic rate. They're not really very motile, so the heart's not real big, okay? We've gone over the adjustments. I will show the movies next time, but I want to take 1 minute to go over studying. Flashcards may be helpful. I got a lot of questions about lecture exam too. I think some students are very disappointed with their score, and they're trying to figure out what they can do to do better. I would highly recommend the following approach. I had one student come on Wednesday to discuss this. I told them they didn't have a lot of time, but they improved their grade from an F to a C on lecture exam 2. So I would recommend you take one lecture. For example, this lecture, try to get on one piece of paper. You do the same thing for Bio 1A. So with Dr. Firestone's lecture, one piece of paper. So basically that's cells and tissues. And you literally want it on one piece of paper. And for every lecture, get Xerox tape, whatever, you want it on one piece of paper. It takes about an hour to an hour and a half to maybe 2 hours to do this summary. But it's very active learning. You take the reader, you take your notes, you combine it into one page. The beauty of this is when you go to study for the final, you have 13 pages. Ideally, you'd summarize those into 3 or 4. What happens is, when you have 13 pages, or preferably 3 to 4, you see the interconnections between the various systems. You see the interplay. So it gives you a good sense of comprehensive overview of things. When you go to study for the final, if you do this for parts 1 and 2, and then the third part, you might be studying 12 sheets. Compare that with 250 pages of notes and reader. It's a lot easier to study those 12 sheets. If you have 5 to 10 minutes, you can get through a sheet or 2. If you have 5 or 10 minutes, you can get through maybe 6 or 7 sheets. But that wasn't very much of the percentage of material. So this way is usually very helpful. I can't guarantee it will work for you, but I might recommend you at least try that. And go back and do it for parts 1 and 2. So good luck and enjoy the lab.